We're going to slowly move now from research, laboratory, and eventually later on to practice. Now to serve as like a pivot point, who else but a person who has who's currently in the academe now in research for 13 years, but he has with him as well 16 years of solid practice, mixed practice, and even swine practice as well. He is a professor at the University of Saskatchewan. Everybody, I think, would recognize him. He's a fixture in a lot of international symposia. He is, his field of expertise is, aside from practice, he is also into research and into discovering emerging swine diseases as well. To talk about and zero in on resilience in reproductive PRRS, let me give you Dr. John Harding.
Thank you, Carlo. It is my honor to be with you in Japan today to present some of our research looking at resilience in the reproductive PRRS model. The results that I'll present today have originated from experimental infection studies of type 2 PRRS infection, and our model uses landraised purebred gilts at 85 days of gestation, which are inoculated with type 2 PRRS. We collect samples from the pregnant gilts from day 0 through to day 21 post-challenge. And then at day 21, we terminate both gilts as well as the fetuses for many different measurements. Over this trial, or series of experiments, we have inoculated close to or over 200 pregnant gilts. And we use a lot of downstream analyses, both looking at virus levels, immunology, genomics, transcriptomics, pathology, and some others, to look at responses both in fetuses and in gilts. To measure or assess resilience in the pregnant female, one way of looking at this is to compare mortality rate, fetal mortality rate, to virus load or virus concentration in the fetal thymus. This is a 2x2 plot that on the y-axis shows virus load in the fetal thymus, averaged across all the fetuses in the litter, and on the x-axis shows fetal mortality rate. And you can see in the top right-hand side we have some of those more susceptible gilts, and this is Gilt Level, which have high fetal mortality rates and high viral load. And by contrast, in the bottom left-hand side, we have some gilts which show low fetal mortality and low viral load. And those are very important gilts because we are trying to understand what are the factors that are associated with those two groups of animals, one being susceptible to infection and one being resistant to infection. These images show the effect of PRRS on fetuses. And we wanted to assess the different factors that are associated with resilience in fetuses. So on the left-hand side, we see viable fetuses. These are all collected at day 21 post-infection in our experimental model. The viable fetuses look normal. They're white-skinned. They may be infected. They may not be infected. But at 106 days of gestation, they look fairly healthy externally. The next stage we call meconium-stained fetuses, and these are fetuses showing early signs of fetal compromise. They're covered with a yellowish meconium staining as fecal contents. They're undergoing periods of stress, and most of those are very high viral load fetuses. The next two stages are dead fetuses, so those that by day 21 have succumbed to virus, and we see some decomposed fetuses, which are largely white externally. They appear to have died within a couple of days of our termination point. And then we see another grouping of fetuses we call autolyzed fetuses, which are fully decomposed and undergoing rapid decomposition. And our assumption in this model is that our viable fetuses are in fact more resilient than our decomposed and autolyzed fetuses, which have died very quickly. And this is looking at the knowledge that the uterus is in fact positive for virus within about 2 days of infection. Now, this image shows those fetuses that may be grouped across the litter. And the positions are represented along the top axis. L1 would be the first fetus on the left uterine horn, and the first position is closest to the ovary. And the same, but on the right side as well. So the R1 fetus would be the first fetus on the right horn closest to the ovary. And then sequentially, as the numbers increase, we're moving those fetuses closer to the body. You can see in the top row, the different rows represent different gilts and how the fetuses are placed in those litters. And the different colors of the squares or rectangles represent the different fetal preservation status as shown by the images on the bottom. The first gilt on the top row is a control litter. So non-infected, all fetuses are viable as would be expected. The second is a PRRS infected litter, and in fact, all fetuses are viable in that litter as well. The third litter, we have mainly viable fetuses with a few meconium stained. And as we get into the fourth litter, we have the opposite extreme. So a very susceptible litter where we have most fetuses that are dead, decomposed, or autolyzed. The second last row represents a litter which has dead fetuses on one side and viable fetuses on the other side of the uterine horns. And then the final litter is one that has some dead fetuses, some live fetuses, and that really stimulated us to ask the question whether death or viral infection clusters within litters. So our overarching hypothesis is to increase, or our goal is to increase PRRS resilience in the reproductive model. And we believe that that comes in different components. And the first is you can either do that by increasing resistance of the dam, and that can be done either through systemic immune responses, controlling the virus, for instance, or it can be done through events that are occurring at the uterus and the placenta. We call that the maternal-fetal interface. Or alternatively, we could increase fetal resistance, and again, that probably occurs either at the placenta level or that could occur in fetal tissues themselves. So we're going to start off by talking about maternal resistance systemically. So this plot shows virus levels in the blood of the pregnant dam from infection through to day 21. And you can see these are all PRRS infected animals. You can see at day 0, all PRRS levels are 0 in blood. By day 2, we have all females are infected and showing high levels of virus in their blood. Virus levels peak at day 6 in this model, and then by day 21, virus levels drop with some females clearing virus from their blood by day 21 post-infection. We see there's a lot of variation in the levels of virus in blood, and interestingly, the levels of virus in blood really have no bearing on reproductive outcome. This is an image, or this plot is showing levels of total white blood cells in the blood of pregnant females. And again, those 4 time points, day 0, 2, 6, and 19 days post-infection. White bars are showing white blood cell levels in control animals that are non-infected. Black bars are showing levels in infected, PRRS-infected gilts. And you can see by day 2, there is a substantial drop in white blood cells by about 40% in infected females. White blood cell levels rebound or start to rebound by day 6, and that's across all the different types of white blood cells. And then by day 19, all of those white blood cells are back to baseline levels. And some subtypes of white blood cells are, in fact, very important in resilience to PRRS, and we'll talk about that a little bit later. This plot shows interferon alpha levels in the blood of pregnant gilts following infection. Again, we're using the same 4 time points, 0, 2, 6, and 19 days post-infection. The blue line shows interferon alpha levels in the blood of control animals, non-infected gilts, where the red line shows interferon alpha levels in infected animals. And you can see that interferon alpha levels peak by day 2 and then drop back to baseline level by day 6. And remember, 2 slides ago I showed you that virus levels peak by day 6. So in fact, it's a bit ironic that we have a drop in interferon alpha, which is a very important antiviral cytokine, at the time when virus levels are very high in the blood. And the reason for this is that PRRS causes suppression in interferon alpha. And we've known that for a long time. Another way of looking at interferon alpha suppression is shown in this particular line plot. And in this experiment, what we did was remove the white blood cells from the blood. So we bled the gilt, we collected white blood cells, and in the lab we re-stimulated them with PRRS virus and measured the amount of interferon alpha that those blood cells produced. Control animals are in blue, and there's little change over the first 3 days at least. The red line shows PRRS infected animals, and you can see interferon alpha levels drop on day 2, drop further on day 6, and are maintained at low levels through day 19. So this shows that we have sustained interferon alpha suppression caused by PRRS virus in the pregnant female. We used multivariable statistical modeling to try to determine the factors that we could measure in the gilt prior to inoculation that may be associated with fetal mortality rate. And there are 3 important factors that you can see. The first is IL-10 levels. IL-10 is a regulatory cytokine, and IL-10 levels are very important and are beneficial. NK cells are innate immune cells, which are effector cells of the immune system. And again, levels of NK cells are also very important in the control of PRRS and offer a beneficial effect. By contrast, interferon alpha levels, which we know are very important, interferon alpha is a very important cytokine that's involved in controlling virus. But what we found in our study was that interferon alpha levels are in fact detrimental to fetuses. And the higher the interferon alpha, the higher the fetal mortality rate, which goes against our previous beliefs. Now, we also looked at which genes present in the gilts were associated with susceptible or resilient gilt. So again, this is a 2x2 plot. And we show on the top right-hand quadrant the susceptible females with high mortality and high viral load. And we're comparing those to the resilient population of gilts, which have low mortality and low fetal load on the bottom left-hand quadrant. And we're measuring the genes or the transcripts present in the blood that are either up or down regulated and may be involved in resilience to PRRS. And what we found is that low fetal mortality in gilts really was the result of these gilts being primed and ready to respond. They have very fast adaptive immune responses. And this is all measured prior to inoculation. So high levels of platelets. Platelets are cells in the blood which repair vessels after damage, and we know that PRRS causes vasculitis. But platelets are also an effector cell of the innate immune system. So we think they have direct effects fighting off infection. We also found that the faster or the higher the baseline interferon and pro-inflammatory responses the gilt had, reduced fetal mortality. We also saw that the gilts with low mortality had faster adaptive and cell-mediated immune responses. So to summarize this first section, we've learned that interferon alpha levels following PRRS2 infection appear to be detrimental to the fetus. We've learned that resilient gilts appear to be immunologically primed and have faster adaptive immune responses following PRRS infection. Now we've learned that levels of virus in sera are unrelated to resilience, but what they do is seed the uterus with virus. So let's turn our attention to what's happening at the uterus. So within the uterus, the fetus is contained within its own placental unit, and that has three parts: the amnion, which circles the fetus, and then the two other layers, the choriontolus and the allantois, which are fused to create the allantochorion. And the allantochorion then is in contact with the endometrium to form the maternal-fetal interface. This is an electron scanning image of the maternal-fetal interface, where the maternal endometrium is at the top of the image and the placenta is at the bottom of the image. And we have the two pieces that are separated, and we see that both surfaces are covered with both primary and secondary folds, and within each of those folds is a complex network of vessels which supply nutrients and remove waste products from the fetus. And the distance separating the maternal and the fetal blood supply at times is less than 2 micrometers, or far less than the thickness of a human hair. PRRS causes apoptosis of CD163 macrophages, and those are the cells in which PRRS replicates. But in addition to that, causes apoptosis of surrounding cells, and these would be innocent bystander cells, and that includes the uterine epithelium and the fetal trophoblast. This image shows the endometrium on the left-hand side and the placenta on the right-hand side, and the red dots are in fact apoptotic cells, so cells that have undergone programmed cell death. And the large black bar that separates the fetal and the maternal epithelium is in fact a small area of placental separation. Now, apoptosis is associated with high viral load in the fetus and the endometrium. It's associated with severity of vasculitis at the interface, and it's also associated with higher risk of meconium-stained fetuses. And the hypothesis is that apoptosis causes placental separation, which then leads to fetal death. But no one has ever proven that. So we've undertaken a couple of studies then in an attempt to try to understand what causes fetal death. And in this particular slide, we selected a number of viable meconium-stained and decomposed fetuses, and we measured placental separation across those fetuses and categorized placental separation as severe, mild, or none at all. And what this slide shows is the probability of being in each of those categories. And we can see our hypothesis was that meconium-stained fetuses would show higher degree of placental separation because they are undergoing fetal stress and compromise. In fact, we did not show that. In this slide, the viable and meconium-stained fetuses show that placental separation is equivalent in those two groups. But the placenta separates associated with decomposed fetuses, and we don't exactly know whether, in fact, the placenta is separating before or after death. So to summarize this section, I'd like to state that PRRS replicates in the CD163 positive macrophages at the maternal interface. PRRS induces apoptosis of those macrophages and of surrounding cells, including the uterine epithelium. The severity of apoptosis is associated with the severity of disease. But although the severity of placental separation increases after death, we're uncertain whether placental separation actually causes death. We also looked at factors that were associated with dead fetuses, or the likelihood of fetal death. And these looked at a number of factors, both occurring within the gilt as well as in the maternal-fetal interface. And there's some very important factors. So in the gilt's blood, the number of helper T cells is beneficial. By contrast, again, levels of interferon alpha, in fact, increase the odds of fetal death like we've seen before. Viral load in both the maternal-fetal interface as well as in the fetus are very detrimental to the odds of fetal death. In fact, is almost essential for fetal death in most situations. But interestingly enough, to answer the question of whether death clusters in litters, we compared fetuses that had 1, 2, 3, or 4 dead fetuses adjacent to it. And what we found was if a fetus had one adjacent dead fetus next to it, either on right or left, the odds of that fetus being dead were 3.6. But if that fetus had 4 adjacent dead fetuses, 2 on right and 2 on left, the odds of fetal death increased substantially. So this shows that fetal death clusters in litters, and we believe that index fetuses become infected first, and then the virus spreads laterally to neighboring fetuses. Now, virus load is very important, a very important risk factor associated with fetal death. And one of the questions we had is whether virus levels differed between large fetuses and small fetuses. So we categorized virus load according to 4 categories shown by the 4 different colors of lines on this plot. The yellow line being high viral load, green being medium, red being low, and blue being negative. On the x-axis in this plot, we represent birth weight of pig by the degree of intrauterine growth retardation. So on this plot, large, healthy, fat pigs are on the left-hand side, and small, runty pigs are on the right-hand side, those that are experiencing high levels of intrauterine growth retardation. And in fact, what we found was that large fetuses have a much higher likelihood of having high viral load, whereas small runty fetuses have a much higher likelihood of being PRRS negative. And this was exactly opposite to our hypothesis, which I would have thought that the runty pigs are always the ones that are most susceptible to disease, but that in fact isn't occurring. So why is this? Well, it could be that large fetuses have large placenta, so there's just greater opportunity for virus transmission. Or it could be that large fetuses have a metabolism that is different than small fetuses, such that it supports viral replication. We don't exactly know the answer to that, but it is an interesting mystery. Now, the study we performed had a lot of fetuses, over 1,000 fetuses that we could collect quality genomic DNA from. And this allowed us to perform the first ever genome-wide association study of host genomic factors that are associated with resilience. And credit for this work goes to a gentleman called Tianfu Yang, who is a PhD student at the University of Alberta. So what we did was looked at several important phenotypes in fetuses and then identified specific single nucleotide polymorphisms, or SNPs, that were associated with those phenotypes. So this first phenotype that we'll talk about is fetal viral load. We understand the importance of fetal viral load, and with our genome-wide association study, we identified 3 SNPs present on chromosome 1, 12, and 14 that control about 13% of the variation in viral load in those fetuses. And those are represented by the blue dots. And red circles. Now, for each of those SNPs, you have one pair, one allele from your mother, and you were given one allele from our father. So there are 3 different outcomes. If you receive 2 A alleles from your mother, then you will be an AA. If you receive 2 B alleles, one from mother, one from father, you'll be a BB. Or if you receive 1 A from one parent and a B from another parent, you'll be a BB. So 3 outcomes, AA, AB, and BB. And this series of plots shows each of those 3 SNPs across those 3 different fetal outcomes. Viral load is measured on the y-axis, and fetal genotype is on the x-axis. So looking at the middle box plot, we see that the BB allele has viral load that is about 1 log lower than fetuses with AB or AA alleles. So what that shows is that the B allele is recessive, and one fetus requires both B alleles to see the drop in viral load. And a 1 log drop in viral load is pretty important. The plots on the far left and the far right show the opposite. So in this case, a fetus needs both A alleles to show the lowest viral load. If they're an AB genotype, then they have intermediary viral load. And if they're BB genotype, then they have high viral load. So these 3 SNPs together work additively in addition to this, meaning that fetuses that have more beneficial alleles have, in fact, lower viral load than fetuses that have fewer beneficial alleles. So this plot is one of our other fetal phenotypes, and this was the likelihood of a fetus being viable compared to meconium-stained or decomposed. And what we see is that there is one SNP present on chromosome 7 that accounted for 35% of the variability in viral load. And this is very interesting and very exciting. And we've got further work underway to try to understand which genes are located near that SNP and whether they biologically have importance for PRRS infection. So to summarize responses in the fetus, pathologic lesions are rare. We didn't even talk about pathologic lesions, but they are very rare, but they markedly increase the odds of fetal death. Fetal death and viral load cluster in litters and likely follows lateral transmission within litters or along the surface of the endometrium. Intrauterine growth retarded fetuses, or runts, are more likely to resist infection than large fetuses. We found 4 single nucleotide polymorphisms that may offer potential opportunities to select animals with increased resilience for reproductive PRRS. And interestingly, none of these SNPs are related to resilience to reproductive PRRS. So what's occurring in the reproductive tract is much different than what's occurring in the lung. Now, some of you may have recently heard that there has been a knockout pig, a gene-edited pig that has been created, where they have done several things, but one that they've done is completely remove the CD163 receptor. And this is very exciting work, and I think it's ongoing work as well. And now there are a number of papers that describe how well this has worked. But certainly if you knock out the entire CD163 receptor, that's one possibility. Or if you just knock out portions of the CD163 receptor, that is a possibility as well. And we can get into the discussion later this afternoon on whether we think this is coming to market anytime soon, but it is certainly looking like it is very exciting work. By contrast, our approach to controlling reproductive PRRS that we have studied is to really understand and exploit the natural variation in PRRS resilience that exists in our populations today. In the last 5 years, we've made great progress in understanding the complex pathophysiology of reproductive PRRS. And in the next 5 years, our goal is to identify genetic biomarkers to assist in selection, biomarkers in blood to preemptively select or predict PRRS severity, and novel therapeutics that, in fact, may help to prevent against fetal infection and fetal death. And really, our goal is to provide tools for reproductive PRRS that can be used for any pig, any breed, and anywhere. Thank you very much.
Thank you very much.